It is my pleasure to welcome Dr. Hobson this morning.
Well, thanks so much, and thank you for coming out this morning, sort of a brisk morning, and I wondered how many people would show up. And I must say that having taught undergrads for the last 2 months, it's a pleasure to see many of my own cohort here in the audience. So that feels good. Now, the first thing you've already noticed is that I don't have a picture of a monarch butterfly here. Don't worry, we're going to get to monarch butterflies in a big way. I did want to weave a little bit of a background story for you before we get to monarchs, okay? Yeah, let's just see here, I just get organized. So here are monarch butterflies. So, we will be talking a lot about monarch butterflies this morning. This picture was taken by a friend of mine in northeast Mexico near Monterrey and near Saltillo. If you know that area, these are our monarchs, and this was taken last fall, and they were en route to their wintering sites down there. So you all know the story. You grew up in Ontario, most of you here, and so you know a lot more about monarch butterflies than most people in the rest of Canada, as it turns out. So we will get to that. So just bear with me, because I want to give you sort of a bit of a story of how we've approached issues of migratory organisms in general, especially small ones, like butterflies, dragonflies, and birds, and so on. So that's the idea. So I'm actually a bird guy. I do like butterflies, and I've really spent a lot of time working on them and other insects and so on, but I have to say the majority of my career has been on birds. So here's a great example to get us interested in the topic of migration. And this is a bird called the bar-tailed godwit. It's a shorebird. It's about this big, has a long beak, long legs, and it does the following, which is pretty amazing. It winters in New Zealand, and it breeds in Alaska, okay? So that's fine, all right? So round about a couple of months from now, maybe 2 months from now, it will embark on the first leg of a trip to the East China area of the Yellow Sea, where there's a big mud flats, where it will refuel. It does this non-stop. Okay, so it's going 10,300 kilometers to get there. It has enough fuel to get there. It has enough fuel in the tank, I like to say. But once it gets here, boy, it has to stay a few weeks. And it has to replenish those reserves and eat on the mud flats there and get ready for the next leg, which takes it to here, which is another 6,500 kilometers without stopping. Now, that's fine, but then after doing all of that and breeding, laying eggs, and getting on with the next generation, it does the most extraordinary thing I think any of us have witnessed in terms of migratory bird, and that is it flies from Alaska to New Zealand non-stop. Okay? It knows enough that when it hits Hawaii, it takes a right turn, and it ends up down here, and guess what? It has fuel to spare. Could you do that? Okay, so that's a fun example of animal migration and I would say how little we know about the mechanics and the biology and so on of these kinds of phenomena, which is really a fascinating area. The other thing we need to remind ourselves of is that any migratory organism only spends a little bit of time in Canada. Understandably, most of our migrant birds right now are sitting down in Central America and North and South America and so on. So they only spend about typically 4 months of the year sitting here in breeding in our great northern forests and so on, like this American redstart. And the rest of the time, like this particular example, they might be in Jamaica or Cuba or what have you. And so there's a great deal of interest now in saying if we want to conserve or manage migratory wildlife, it's not enough to only know what's going on in our own backyard. We have to know what all these connections are that link it between these different life stages and so on. It turns out that events that can occur here can have profound, if you like, carryover effects or ripple effects on different life stages. That goes for monarch butterflies, it goes for just about anything that migrates. But it's taken a while for people to get into that mindset that, we've got to worry about the rest of the other 8 months of the year. Okay. Well, I'm going to get to monarchs, don't worry. But just giving you a bit of background on the bird side of things. Well, how do we mark birds and study migration? Well, we put these little flags on their legs, or we put these patagial tags on the wings. But more recently, we've been getting really clever and putting satellite transmitter tags on their collars like this, or relying on people now. This is a great time because in social media, people who go out and photograph things can post their pictures up on the social media. And actually, you can start piecing together migratory routes by just, asking people to post their pictures, if you like, the photographers out there and so on. Okay, so that's what we've done, and then this is the gold standard of measuring movement of anything. Here's an osprey, which we put a satellite transmitter on, and we can tell its exact routes. I mean, within 30 meters, we know where it is at any given time with these technology today. So this is all very cool. But, as you probably guessed, you can't do this very often, and you can't do this on very many individuals. It's extremely expensive, and so on. More recently, we've got really clever about putting these little geolocators on, in this case, a barn swallow. Barn swallows have just been achieved the status of being threatened in Ontario. This is a bird that I'm sure in your youth was extremely common. Now have diminished a great deal, so that's an interesting aspect. This is a little device that records day length. That's all it does. It just tells you light level with time. It's a little watch crystal in there, and there's a little, device that does that. And then when you catch the animal a year later, when it comes back, hopefully, you download all that information, and it tells you what the day length was and the time of midday throughout the year when this thing left. With those two pieces of information, you can get latitude and longitude. It was the way the mariners from Europe navigated their ways across the oceans. The big breakthrough was having a clock, so you can tell longitude with the time of midday. And relating that to Greenwich Mean Time can give you that kind of information. So people have been getting incredibly clever with this. This is a study we've done with swallows across the country. Turns out that the West Coast swallows don't go very far. They go down to Central America, but from Ontario and into the Maritimes, they go way down to these kinds of areas. So you can get these amazing bits of information with time. Now, this is an amazing part of the world. We're actually the envy of the world because we have this system that was pretty much made in Western, the university here. It's called MODIS. It's a wildlife tracking system, and I don't know if some of you may have seen these, but there are these antennae that receive signals with these great big solar panels to run them and so on, and they're all over the place. And so if you put a little tiny radio transmitter on birds here, you can pick them up in this big array of these devices. So we're the envy of the world. I mean, they don't have anything like this in the United States yet, or in Europe, or what have you. So there's a wonderful situation here. And guess what? This was really pioneered by the people at Western, as well as Guelph and a few other places as well. So that's what they look like. Put a little radio transmitter with a long aerial. It's just coming down here with a battery on it, and you can see where these go. Well, we've done this in South America with thrushes, and I had students who were working in northern Colombia, and they were really interested to know whether they were sitting in shade coffee plantations or forest and what they did. And then the study was over. They all came back to Canada. But this was sort of the gift that kept giving. These transmitters kept moving and kept going, and we were able to pick them up all the way back again up into this part of the world. So the technology is getting there, and it's getting very interesting. But overwhelmingly, we rely still on leg bands, where we'll put bands or marks on any kind of wildlife and hope somebody else picks it up somewhere else to tell us where they go. So to give you an idea, 3 million Canada geese have been, there's more than that now, this is an old number, 3 million Canada geese have been banded with about a 20% recovery rate. That's a lot of band recoveries. So you can do a lot with these birds. We know a lot about Canada geese, okay? We really do, more than we ever wanted to know. And here, though, is an American redstart, and this is pretty impressive. 800,000 of these small birds have been banded in North America, and many just down the road at Long Point Bird Observatory, Bird Studies Canada, doing this. But their recovery rate is just vanishingly small. can't do anything with those data. So for very small organisms, it's a big problem. All right, so I told you I'm getting to monarchs. We can put tags on monarchs too, and many of you will know about this program, a Monarch Watch. You can even put little transmitters on bees. Now, you know, I've been told that a bee can't fly anyway for various physical reasons, but when you put this thing on its back, how can it move? I mean, you know, this is amazing, but there's a little radio transmitter that people have put on bees, so okay. And on a dragonfly. And Martin Wakelski here is supergluing a transmitter onto a dragonfly, then he goes up in an aircraft, and he's tried to follow these things. So this is all neat and cool and very interesting, but I think you can appreciate we're not going to get a huge amount of data in doing this. This is my colleague Chip Taylor from the University of Kansas. He's the very man who started up a program, if you go to the website, it's called Project Monarch Watch. And they're fantastic. If you go onto their website, they'll tell you how the wave of monarchs is coming up from Mexico and, you know, when is it going to arrive in southern Ontario or Quebec or so on. But one of the things he has done has been pioneering putting these tags on monarchs, these little like stamps, this sticky device you put on a monarch wing, and they can handle it, and they do so. But then you're left with this problem. Because down at the wintering sites, and I'll be showing you a lot more pictures of the wintering areas of monarchs in a little bit, but there's a lot of overwinter mortality, and it turns out that a lot of them don't make it. So really, you can go down to these sites and pick up masses of them that have died over winter, and then you can just go through one by one, and you can look for these tags, okay? And that's what he's doing here. And I actually was at a meeting with him last week, and he said there's 14,000 butterflies in that pile. 2 of them had tags on them. So you can see the problem. And then they've sweetened the pot by giving 20 pesos to anybody in Mexico who can pick up one of these things and find a tag. So, okay, we can play these games and so on. But here's the question. Are there other ways that we can track migratory wildlife that doesn't require us to put tags, any kind of tag, on an individual? So that's the case with small birds. Is there something in the feather? Is there a mark? Is there a fingerprint? Is there some information we can get about origin? The same with the wings of dragonflies and monarch butterflies and painted lady butterflies from Europe. All of these organisms we have worked on now using what we refer to as biogeochemical markers. And I'm going to explain that at great peril to my reputation, I'm sure. But we will get into this briefly. Now, the advantages of using what we'll call an intrinsic marker, that is something you're carrying around inside of you right now, is that it's not biased to origin. It doesn't mean that you have to go out and mark a certain population. They're already marked, okay? Only one capture is required. You only need to sample them once. You don't have to get them coming back or recovering or all that good stuff. And then we can typically get information on thousands of individuals rather than just a handful that you could put these very expensive pieces of equipment on. And, you know, I think this is a big one. It doesn't influence their behavior, and they're not carrying something we've stuck on them, okay? So this was the motivation for doing a lot of this. Now, I promise you, this is the only real chemistry slide in the entire talk, okay? So if we can get through this slide, we'll be fine. It's all downhill after that, all right? So bear with me. If you remember your first year chemistry or whatever, elements occur in more than one stable form. There's a very common form, which is the lighter form, which has the atomic masses here. This is basically based on their weight, their mass. And then there's a slightly heavier form. So in the case of hydrogen, this will have an extra neutron in the nucleus, makes it heavier. Okay, doesn't matter about that. This form is heavier than this form, but this form only occurs in way less than 1% of all hydrogen. It's the same with most of these. They're very rare, but they do occur, and they're stable. They don't go anywhere. So we depict this with this weird notation here relative to a standard. Don't worry about that. These are just numbers. But what we're really interested in is the relative abundance of the heavier to lighter isotope. So I could take a hair sample from you right now, run it through a mass spectrometer, and I could tell you what the ratio of carbon, heavy carbon to light carbon, heavy nitrogen to light nitrogen, et cetera. We could play that game with a number of tissues and so on. All right. That was the difficult part. The easier part to understand is that if we look at the world, it has all kinds of patterns of these isotope ratios. They're not constant. They're not constant from one place to another, but they do follow some rules. And they especially follow rules when it comes to plants and the photosynthetic pathways. The type of photosynthesis will really change that carbon isotope ratio. In the oceans, we have very different patterns on nitrogen and carbon isotope ratios, and so on and so forth. Now, the interesting thing is that, you just take this for granted. I'm just telling you this occurs. These patterns, like South America, this is what the vegetation would look like in terms of their carbon isotope ratios and some kind of index, but they're very different in the Amazon than they are in the grasslands, for example. Well, we've known about this for 40 years, 50 years, longer than that, actually. And so it wasn't actually until very, very recently we asked the question, well, if a bird is living here and growing its feathers here, and then it moves over here, does it take that signature with it in its body? And if it does, especially if it grows structures that you can sample, like feathers or hair or toenails, could you then infer where it might have been, 2 months ago or a little while ago? So this is the kind of base map fingerprinting technique that we've been getting into over the years. Again, I will get to butterflies, okay? But you're getting a real spectrum of taxa here, which I think is kind of neat as well. This is work that was done, I showed you a pattern just recently where the food webs in the oceans can have these very different signatures, and they stay, faithful to those signatures. And here's an animal, the bowhead whale, that the western population migrates from the summer from the Mackenzie River estuary and the Beaufort Sea out to the Chukchi Bering Sea and back again. This gets ice covered, of course, and it's just moving back and forth. It raises its young here and it goes over here, and it doesn't go very far. It does that on an annual basis, okay? All right, well, that's cool. And then they grow these lovely big baleen plates, which are used for filtering zooplankton out of the water column. And they're like your fingernails or your hair. They're continually growing. But once they're formed, they're inert. They don't change. And they lock in these isotope signatures. And so this is what the picture looks like. We've got some kind of base map where these isotope signatures are changing in the food web, and we've got an animal that's moving back and forth with a continually growing tissue. And these are the patterns we get. These are for those carbon isotope ratios, these are for nitrogen, but these are the lovely sinusoidal patterns. Guess what? They refer to those annual cycles. They're moving back and forth. Now, when this was first discovered by Don Schell in Alaska, people said, you're crazy. You're crazy because we know that that animal that you sampled must have been X number of years old. And he said, well, no, it can't be that old because, or it must be older, because we've measured these things and we know that these correspond to that annual event. And so this really shook up the world because the people who were studying whales before were using whale length as some kind of index of its age. So with young animals especially, they would say, that's 7 years old or that's 15 years old. From aircraft, you can fly over them and you can get an idea of how long they are. It was all wrong. It was all wrong. So they had the whole data based on the maturity of these whales was totally wrong. And he was able to show that with this elegant kind of way. Africa, elephant ivory. This is nothing new. Chemists have done this for years. These are strontium isotope ratios and nitrogen isotope ratios. But all I'm really showing you here is national parks in Africa. And this is the ivory from those parks. The idea was that you could fingerprint ivory. You could find a piece of poached ivory and you could say, that must have been coming from this park versus this one, and so on. This is ongoing. There's a lot of interest now in still using this to fingerprint tusk and so on. But I've worked a lot on birds. It requires doing a lot of work in captivity and figuring all these things out and putting them in wind tunnels. I don't know if any of you know that Western is the only university in Canada that has a really state-of-the-art wind tunnel at the Afar facility where they fly birds in these very elaborate devices and they can make them exercise and so on. So right here, there's been some really cool work done on this, and we've been playing those games too. You can look at anything. You can look at feathers for those isotope signatures. You can look at toenails. You can also look at breath, okay? Now, it turns out that if this bird feeds on lovely sugars in fruits, like berries and so on, its breath will tell us that that's what it's been doing, as opposed to feeding on insects and so on. All right, well, that's neat. How do you get breath out of a bird and put it through a mass spectrometer? If you're interested in that, I'll tell you about that later. What about polar bears? Okay, this is a polar bear blowing up a bag of breath. It's a one-way valve here. This is a mask over it, and it's just cooperating. It's not drugged. It's just well-behaved. No, it's definitely been immobilized. Now, here's the story. When polar bears in the western Hudson Bay population are forced on land for longer and longer and longer periods now because of the melting ice, it's of interest to us to know, can they nutritionally make it? What are they feeding on? Well, it turns out they eat berries. So polar bears will eat lots of berries on land. So we were interested to know from their breath, are you really eating a lot of berries? Are you metabolizing a lot of berries or not? It turns out they're not. It turns out that they're doing it because they've got nothing else to do. They're not too distant relatives of brown bears that feed a lot on berries, but these guys have departed, and they put on lots of fat from seals in the winter, and then they mobilize those stores for their energy. What they're getting from the berries is not going to help them through these longer and longer periods of time. So that's why we were collecting breath on these guys. I didn't get to do any of this neat work, by the way. These guys get the breath, and they send you a little tube of breath, and looks like anybody else's breath, and so on. So. Now, the huge breakthrough, and maybe some of you don't realize this, but the really huge breakthrough for animal tracking is becoming from precipitation, from rainfall and snowfall. It turns out that there's information in precipitation that we never imagined could be used to the degree it has. And I'm just going to go through that very briefly with you. It turns out that the hydrosphere is very interesting. That is, where do we get water from, and where does it end up, and where does it go? A fascinating area of hydrology and research. But guess what? When you get evaporation from oceans to make clouds, and those clouds rain out, that really does affect those isotope ratios in the water. Well, what's water? H2O contains hydrogen and oxygen. That hydrogen has different isotopes. The ratios of those two very much change through all these kinds of processes. I won't go into it any more than that, other than to tell you that if you know about that stuff, you can really start to put a map together. Now, the International Atomic Energy Agency, right after the Second World War, started sampling rainwater all over the world. These are all the stations that they have been using to sample water. And what do? They take a container that's been the accumulation of rain over that month, and then they send it off to a lab, and then they get the next month, and the next month, and the next month, and so on. So they've been doing this for 60 years. Does anybody know why you would ever want to do this massive effort for rainfall? It turns out that we were really worried about people blowing up atomic bombs and atmospheric bomb tests during the Cold War. And in those days, the way you could tell whether China or France or somebody else had blown up one of these devices was just simply measure rainfall. Because a lot of radionuclides went into the upper atmosphere and then they get rained out, and you can say, oh, wait a minute, our tritium levels or our radioactive hydrogen levels have gone up suddenly. And they look at wind patterns and where the weather's coming from. And they were using it as a way of determining who was behaving themselves in terms of blowing up atmospheric bomb tests. They still do it today, but they're doing it way less for that reason, because they were also measuring the stable isotopes of hydrogen as well, but they didn't care about those very much. It turns out this has become a gift to us looking at migration. Why? Because there's beautiful patterns across continents of these isotope ratios of hydrogen in rainfall. Okay, that's all you have to know. It looks like this in North America, which we have one of the best situations in North America, because we have these lovely gradients. Little weird in other parts of the world and so on, but we'll talk about those. So here we are in North America, down in Texas. The rainfall has a lot more of that heavy isotope of hydrogen in it than it does up here in London, and certainly way more than up in the polar regions. But it kind of spreads out in these lovely latitudinal bands. So back in the 90s, a colleague of mine who studies this kind of stuff in Saskatoon and I got together, and we just sort of said, well, if we're seeing these patterns, surely they could get transferred up into organisms through the food web. By the way, all of your hydrogen in your body right now comes from precipitation. That's where it comes from. It comes through the food web, through plants. Plants take up that water, they fix that hydrogen in plant material, animals eat that, et cetera, et cetera. No matter if you're a carnivore or a vegetarian or whatever, it doesn't matter, you're getting all of your hydrogen from there. So we thought, well, let's try this. So let's go across the continent, let's sample birds that we know grew their feathers all across North America, and let's just test it out. And this is the pattern we got. This is the feather isotope value with the rainfall isotope value. And you could say, nah, that looks pretty noisy, you know, that's not very useful. It is. I mean, to us, this is pretty remarkable. These are from the north, in the polar regions. This is from Texas, and everything else in between. And this is the basis now of us using this technique to take an animal and put it back onto the map of where it may be coming from. So it wasn't long after we came out with that work and proved that it worked that a number of people published in some pretty prestigious journals. And what did they do? They just went and grabbed feathers of birds. This is the black-throated blue warbler. This is its breeding range in North America. This is where it winters. And they found that the ones, and by the way, these birds grow their feathers before they go down here. They keep them till the next year. So this is where all their signal is coming from. If you sample them down here, you can ask, where did you come from? And this is the pattern they got. The northern ones were wintering in western Cuba, and the southern ones in the more Dominican Republic and the Eastern Antilles. Okay, that's interesting. But guess what? These are stable. These populations are reasonably stable. These are declining heavily. So we think that's probably because of something going on here rather than something going on here. These are the Appalachian Mountains and so on. So you can see that we can start playing these games. The same thing was done with museum samples, feathers taken from museum skins of these birds, and they found that the birds that breed up in Alaska winter way down here. The birds that breed in southern California that don't go very far. This is called leapfrog migration, as you can imagine. So suddenly, people went crazy, and they could apply this technique to all kinds of things. And suddenly we got information we could never get before. We could go down there and place birds from Guatemala in the wintering grounds to very, very specific places on the breeding ground. Now, I am getting to monarch butterflies. In fact, you people will be patient enough, but I think you'll find this still interesting enough. You remember the Hudson River flight accident where the plane went down and landed on, and nobody was killed, and the whole thing was a brilliant piece of news. Well, the plane was brought down because Canada geese went into the turbines of the aircraft, and feathers were left. And so some colleagues of mine took those feathers and they looked at the isotope ratio of the feathers, and their big question was, were you from New York City? Because if you were from New York City, you are in trouble. Because nobody wants Canada geese in New York City on the golf courses, and it's like here, right? People don't like these things around, and if they're bringing down aircraft, wow, we're going to get rid of you right now. So that was the big idea. Are you a local Canada goose? And if you are, that's going to be a big deal. Well, it turns out that, just to give you this is the ones that brought down the U.S. Airways flight. It turns out they're from way up in the north. They're not from New York City. The New York City residents are up here. These are the guys that brought the plane down. Okay. It turns out that the authorities in New York are still trying to get rid of their geese. But this was a lovely little forensic story we can use. I'll just finish off some of the birdie examples before we start talking about monarch by telling you about Europe. Europe has a pattern too, but it's not quite as lovely as the one we have in North America. It gets very noisy, but that's fine. We can do that same thing of collecting bird feathers all over the place and getting lovely patterns. And then the French government came to us and said, you have a technique that can tell us where birds are coming from. Okay, I get it. Now, we've been criticized forever because the wood pigeon in Europe is a big pigeon. That's a really big pigeon. And it tastes really good. And it turns out that the French, there are 7 million killed in Europe a year. 5 million of those are killed in France. Okay, so the French are taking by far the absolute majority of these birds. They breed all over Eastern Europe and France, and they winter in France and are hunted heavily in France. So everybody in Scandinavia was saying, stop shooting all of our pigeons. that go and spend the winter in France. You guys really have to figure this out. So the nice thing about hunted species is you can get lots of samples. You can just ask hunters to give you a feather from every bird they kill. And so we looked at thousands of these things. I won't bore you with the details. It turns out all the birds we looked at, most are coming from France. So everybody has a big sigh of relief that the French were not doing bad things in the name of gastronomy, etc. However, if you look at the same species in Corsica, which is an island here which has no breeding wood pigeons on it, Italians by the boatload go there to shoot wood pigeons in the fall, in the winter. Then we can ask, where are you coming from? We know you're not coming from here. Turns out they're coming from this part of Eastern Europe, using that mapping isotope technique that I've been showing you. The other last thing I'll tell you about this is that we can do really cool things now with genetics, which we can get from the base of a feather and the vein that we can get from the tip. This is where we get all of our isotope information. This is where we get all of our genetics information. We're now putting the two together to get a much better idea of where things are coming from. Okay, so that's in a nutshell what we do. To just reiterate slightly, we're using feathers which lock in these signatures much like whale baleen does, because they only grow with feathers at a certain time. They carry them year-round. That signature's locked in, and we can work backwards to see where they're coming from. All right. Monarch butterflies. Now, this Omar is a colleague down in Mexico. So these are a few pictures here we have from him. They do have monarchs in Mexico that actually are resident, that are non-migratory. They actually have milkweed down there, and they have monarchs that use them. But he's a great photographer, and so I thought I would show you a few of his lovely pictures and so on from that part of Mexico, and so on. So these are exquisite kinds of structures that you all know the life history of monarchs, of course, of going from an egg to a larvae caterpillar stage, feeding exclusively on milkweed plants, acquiring toxins that they can use to prevent predation on them. A great story of them getting chemical defenses through their milkweed plants, cardenolides, great, really horrible things, but they're able to store them and in their tissues and so on. And anyway, he has some really lovely pictures. Now, you've seen these kinds of pictures in southern Ontario. These are actually taken in Mexico. These monarchs are doing the same thing they do here en route down through Mexico, etc. I guess the difference is they have different vegetation and a nice blue sky, which we often don't have here. Okay, so let me tell you the story. When we were first starting off this technique on birds, we thought, we could apply this to anything. There's nothing special about birds. Monarch butterflies inherit the isotope ratios in their wings from the larval stage. So it's from a very pinpoint place on a very specific plant from a very specific, part of the world. Could that information be, uploaded into the wings where they keep? If we went to Mexico, could we take these wings and then ask the simple question, where did you come from? Did you come from southern Ontario? Did you come from Texas? Where did you come from? So that was the idea. And for those of you here who don't know the life history of these species, there are two big populations. The western population in California mostly winters in these, eucalyptus groves and western California. And then there's the one we're familiar with here is the big eastern population. This is a very small population compared to the bulk of what we get here. Now, let's just start off in Texas, where they overwinter, Mexico, where they overwinter. It's only the gravid females, it's only the females who are pregnant with eggs, fertilized eggs, that leave the overwintering site in Mexico. So a very small percentage of the ones that actually overwintered there get back as far as Texas, where they encounter milkweed, they lay their eggs. 6 weeks later, you have an adult monarch butterfly emerging from that event. Now that generation of monarchs keeps going north. They don't stick around. They're in search of more milkweed with less competition, if you like. So they think, if we keep going north, we're going to find milkweed up here that's going to be really great for us to reproduce. So these are very young animals. They have a very short lifespan. And so they have to get their reproduction done in a short order of time. So 6 weeks from the egg, they're back as an adult, and they're moving north. They get up to about here, and then there's another one and another one. Well, the ones that reach southern Ontario could be 4 or 5 generations removed from the ones that left Mexico, depending on weather conditions, depending on climate, depending on whether there's lots of milkweed that year or not, whether they overshoot or come all the way up in one flight, very rare. That would probably never, almost never happen. The ones we're getting were born in the US, and then they come up here and do the same thing. And then, as you know, in the fall, they all head south again in one big migration movement to the Oyamel fir forests of central Mexico, which are these high-altitude fir forests. They're at about 3,500 meters typically, so it's very cool, it's very stable in terms of the climate, and these butterflies have learned that that's a great place to be. All right. Now, as you know, monarch butterflies, maybe you may know this, they're not doing very well. We had periods where I was working on them in the early 90s where we had very large populations, and now they've dwindled down to very small populations. There's a lot of interest now, a lot of concern from 3 countries, US, Mexico, and Canada, of what may be going on, and so on. Oh. And so we said, can you get around this tagging thing? Because it's not really telling us where the bulk of these monarchs are coming from and going through this process. It's just not. It's neat, and I'm glad people do this, don't get me wrong. It's just not going to answer the question. So what we did, we got elementary school kids to put eggs of monarch butterflies on milkweed in their schoolyards across the range, the eastern range of the monarch butterfly. So you know the story, and I'm sure many of you have done this. You've raised monarch butterflies on milkweed to adults, and it's a beautiful story, and it's a great part of the education of people in this part of the world. Certainly in Saskatchewan, nobody does this because we don't have monarch butterflies, so it's a real Eastern Canada thing. So we sent out eggs to all of these schools, these elementary schools across the US and Canada. So we had 100 or so schools that were participating. What they would do is they would go outside, they would put the eggs on the milkweed, and then they would generate a cohort of butterflies. We said, you can do that, you do whatever you want, you cannot water the garden. You cannot artificially irrigate the garden. You have to rely on natural precipitation, please. And they did that. So they were young scientists who were doing this wonderful project, and they raised these butterflies in their backyards without watering the garden. Why wouldn't we want them to water the garden? Because we wanted this to represent a base map which would be natural, what would be the natural precipitation driving this. Why did we do this? We wanted to generate a base map to say, this is what it looks like in this year for where these monarch butterflies should be coming from. Okay, so that's what they did. They sent us those butterflies, and we analyzed them. And we came up with a picture. This is an old picture, right? Because this is a Cree surface, and this is going back some years. You can get much more fancier pictures now. But we have a lovely 2-isotope base map. All you have to know is that we have carbon and hydrogen. We're showing a lovely pattern, and we could then give you with some reasonable confidence where those monarchs are coming from if you go down to Mexico and try to work backwards. Just say, where were you born? Where were you born? And so on. So that was the whole idea of that in the 90s. So the next part of the story is after we got the base map from those elementary school children across the country, we went down to Mexico. And in those days, there were 13 known roost sites of monarch butterflies. Today, there's about 5. Okay, so even in my lifespan, even in the 25 years I've been working on monarch butterflies in this story, their numbers have gone way down, so we have very few now. So I'm really pleased we did what we did in the early 90s, because it really was a valuable data set. Now, it's great. You leave Saskatoon and then you go down to these 3,500 meter locations of these fir forests where these monarchs are, and I can tell you that if you're not used to low oxygen levels at high altitude, then this was tough. You know, pounding headaches and all that, where everybody else around you is just miles ahead of you. So it was a silly thing in many ways. But we managed to go to these. Many of them are very commercial, not commercial, but they've been very well established, where the local people who own the land are running these monarch butterfly roost phenomena. It was a Canadian, Urquhart, from University of Toronto, who first established where these things were. It was unknown to Canadians, or North Americans at least, Mexicans or North Americans. But what I'm saying is, we didn't know where they wintered. They just disappeared from here. And he, in the 60s, found where these sites are. And now, the time we did it, we went to every known roost site. So some of these are very well established, got all the washrooms and whatnot. And here's where you go. You just follow the sign to go to where the monarchs are. And as you approach those wintering roost sites, you start to get a feeling of something's happening. You start to see monarch butterflies, and they start to be very, very present, flying around, and you think, we're close to something here. So it's a very exciting thing to experience. There are millions of butterflies that are congregating from all over North America to these sites, and they're literally covering these roost sites in that part of the world. And then you go to the roost sites itself, and they look something like this from a distance. Now, these are big hanging masses of monarch butterflies. And these trunks are covered with monarch butterflies. I'll show you some better pictures in a moment, but from a distance, you see this, and it's sort of like, wow, what's going on here? And I want to ask you, how many butterflies are in that picture? It's actually very difficult to get how to quantify how many butterflies there are. The way they do it is they don't even bother trying to count them. What they do is they take GPS coordinates of the points around the number of trees that are being used, and they say, the area this year is this area. So that's the area of the colony, and they're really doing a very crude estimate. All the population estimates are based on area of the winter site. Anyway, so as you get closer, a little bit more color and things happening. But again, a spectacular thing. Some people in the audience have probably been to these sites, the ones that are accessible to the public. And during the day, it's great to get there in the early morning because everybody's cold, and they're just sitting there. Then as the sun hits the angle of these colonies, which, by the way, any given roost site down there would be no bigger than this auditorium. We're talking about 15 trees, 20 trees, masses just congregating in those areas. And then over the winter, they move down slope. And what they're doing is they're tracking the angle of the sun. So they are actually moving as a group, and they leave a trail of dead monarchs behind them so you can tell where they were, and so on. Why are they dying? They don't have enough fat. They don't have enough fat to last them through the winter. They have to accumulate fat because they don't feed on these sites. They only drink water. So all of the sites have to be near a creek or some sort of water body. So they need water, but they don't need food. And there's nothing there to really sustain them. So they're just hibernating, or they're just hanging out. But as the sun hits the trees, they start to quiver and warm up, and they can fly around once their body temperature gets up a high enough temperature. And what they often do then is go and look for a place to drink water. So it's kind of fun. You can go to just watch them all landing at a site. I'll show you that in a minute. And then they go back and roost at the tree again, and so on and so forth. How they find these sites is a topic of another lecture. But anyway, these are the kinds of things, and these are drinking in a puddle. They're drinking water on the ground, and so that's their daily routine. They'll do that. And then in the spring, right about now, a couple of weeks from now, they will start mating. You'll start to see couples forming. And those mated females will then try to do the arduous migration back again to Texas. And the others, well, we don't know what happens to the others, but they don't really factor into it from an evolutionary sense. So that's their kind of story down there. So what did we find? We found that over 50% of the population was coming from this region. Very little coming from Canada, understandably. We're right at the very end of the route. So we're producing monarchs, but we're not producing the most monarchs. We get about 10% coming from Canada, 50% from the American Midwest. Now, why was this? This was a stunning piece of news when we first came out with it, because nobody knew where the big production zone would be. And then suddenly they would say, well, wait a minute, this is the Corn Belt. This is the big Corn Belt region of the US where all the corn is produced. How can you have monarch butterflies coming from such an agriculturally intense area? Well, it turns out that even in those cornfields, you do have milkweed. In Iowa and some of these states, you have milkweed growing in association with the crops and so on. Very interesting. The work that's been done since then with us has been showing very much the same thing. This is a given year, but it's all showing these corn belt regions really producing most of the monarchs. This is a much more recent look over many, many years now, and that region is always showing up as the one with producing the most monarchs. So we've repeated this now 20, 30 times, and that's the story. And why then did this cause such shockwaves? It caused shockwaves because, guess what? Monsanto loves the American Midwest because that's where it uses all of its genetically modified corn. They used a corn which produced Bt, which kills larvae and so on, but that's beside the point. We're not really worried about that. What they've done is they've created genetically modified corn that they can spray herbicide on. Okay, so these genetically modified products, you can spray, Roundup on them, and you can kill all the weeds, but you don't kill the corn. So the corn is impervious to that problem, but it's served to wipe out the associated milkweed that was growing in those sites. Okay, so the price we're paying for high-intensity agriculture with genetic modification is that the monarchs are losing the vast majority of the milkweed that was producing the vast majority of their populations. Now, I'm not saying that the Midwest is still not the most important place. It actually is. And now the milkweed is now found along roadside edges and so on. But it's had a tremendous impact on the population. Why was this fascinating to the Mexicans? Because the Mexicans were getting a little fed up of us telling them, you're the problem. You're the problem. You should stop logging these winter roost sites, and you better look after our monarch butterflies. They got a little fed up with that line of thinking when this study came out, because they were saying, well, wait a minute, what are you doing to help monarch butterflies in North America and their breeding grounds? So it's a political, issue that's of interest. Here's some pictures, by the way, of milkweed growing in corn. It can grow under the canopy of corn. Corn actually produces a nice sort of like tropical rainforest canopy for milkweed, and milkweed can grow underneath corn quite readily, and it has been until the last 20 years or so. So we think that this has had a big impact on the population and so on, and it has all of these political ramifications and so on. I'll just give you a few other examples of kind of neat examples of what you can do if you can use isotopes to tell where butterflies are coming from. They suffer a natural parasite. We'll call it OE. It's an ectoparasite that has these, spores on the outside. They accumulate on the bodies of monarchs, and it doesn't, they really suffer when they have big infestations of this parasite. It's natural, but there it is. It turns out that in the American South, people have tried to help monarch butterflies. So the way they do that is to plant milkweed in their gardens, okay? So they're planting milkweed in all of these places in Florida and all the way down there, and they think, well, this is going to help monarchs, right? Because they need milkweed. And indeed, that was a great motivation. The problem is the following. They've been planting the wrong kind of milkweed. They've been planting tropical milkweed. So what happens with tropical milkweed is it flowers year-round. The milkweed we use, and the native milkweed to these locations, senesces during that period, and it drops out of the picture. So when the fall comes, the monarchs are on their way to Mexico. They see the milkweed, they're not interested. They are just interested in migrating. But if they come across milkweed that is flowering, they get a little bit confused. And they think, oh, maybe this is a place to stop. And the diapause is the term used when they go out of their reproductive phase and into the migratory phase. But then they can get fooled. And what we have seen in these green spots are non-migrant populations that have evolved in the last 20 years of people planting milkweed in their backyard. So is it a good thing? Is it a bad thing? Well, let's look at it a bit further. The neat thing about the isotopes is you can take two butterflies and you can say, you're a local, you're a migrant. You came from over here, you guys are local ones. That's easy to tell. We can just run those through the machine and we can tell that for the first time. Otherwise, you would have had a clue. And it turns out that the ones, the proportion that are infected at these year-round breeding sites are huge for the residents. Those residents are really suffering. They're not able to escape this parasite's life cycle because they're there year-round. In some ways, migration can help populations when it comes to disease and parasitism because they can escape that cycle. They can get out of it, and then the parasite or the disease doesn't do very well. So actually, migration could be not a strategy, but it's certainly one of the benefits is you don't have these problems. So that's what we see here. The residents have way more of these parasites than the ones that were fooled into breeding there. These are migrants that arrive and they go, I'm not going to Mexico, I'm going to stay here, this is a great place to be. But they paid the price, they got all these more parasites. And the ones that were just at stopover sites, the ones that were still going to Mexico, they have very low incidence of the parasite. So we're getting a lot of mortality of these things, and people have called these things ecological traps. So the butterfly unknowingly reaches this unusual event in its evolutionary history, and it gets fooled into believing this is a great place to stop. So, as you can appreciate, people have said, oh, wait a minute, please plant milkweed for monarchs, but make sure it's the seasonal, not year-round one. Okay, so get it right. Stop buying tropical milkweed and planting it in your gardens. Plant the right kind of milkweed. You can imagine this is a great public relations problem because you don't want to dissuade people from the good intentions of trying to help butterflies and so on. I would just say that, I'm running out of time, I know, but I would just say that our future work at Western is using those same isotope techniques now, not to look at where they're from, we've got that all figured out, looking at their wings. We're actually now looking at the issue of where do you put on your fat? Where do you accumulate the nutrients you need to overwinter at your wintering sites? So we're using those same fingerprinting techniques by doing some captive studies where we allow them to feed on nectar, and we can see how that signal is translated into the bodies. And we're hoping to say, are there places in northeast Mexico that are very important for monarchs for putting on that last little bit of fat they need to get them through the winter, or is that all done in Texas? Or is that all done somewhere else? And how will that vary with climate change? Okay, you could do the same with dragonflies. I'm going to skip this. This is a neat story about dragonflies in India, and it turns out that they had the longest migration going through West Africa, 14,000 kilometers, multi-generational patterns that we've actually done with the isotope work to show that this is the case. I just wanted to leave you with a couple of slides that bring this home a little bit, maybe, and that is that isotopes can be used for all kinds of forensic tools, not just wildlife. Cocaine is a plant, and a plant grows in very different conditions. This is all cocaine from Colombia, but it has very different isotope signatures. This is the ivory picture that you saw with the elephant ivory tusks, but this is fingerprinting it to source. The US currency is based on cotton. Cotton is a plant, comes from one very small plantation in the US. You can use carbon and hydrogen isotope ratios of the paper of currency and tell if it's fake or not. I mean, there are other ways of doing that, but you can easily tell using isotopes. And then water. People spend a lot of money on exotic water, bottled water. And some of that water they claim is from some special spring somewhere. And you can spend a lot of money on that. And if you run that through the mass spec, it's like, no, it probably just came from the tap just down the road. And people have published papers on that. The other, this is the very last example, I promise you. People in Utah, my colleagues down there, decided, it's a neat something we can do in the summer. We'll put my grad students in a car and they can travel all over the US. And when you see a barbershop, you just go to the barbershop and I want you to just pick up hair off the floor and then take a little bit of water from the tap. That's all you have to do. Just walk in, introduce yourself, and then do that. Okay, so they did this for hundreds of places in the US. And then they just simply looked at what's the relationship between the hydrogen isotopes in the hair and the tap water. And it's better than birds. It's better than monarch butterflies. It has this kind of amazing relationship. So humans are incredibly well hydrated. You drink a lot of water. Whether you drink coffee or tea or beer, it doesn't matter. You're probably the one species on Earth that has the most water through your bodies. It turns out that really links you with all your tissues to local water sources. So that's what's being used now forensically, because guess what? We can create a human hair isoscape. I've shown you isoscapes of butterfly wings, but this is what it looks like for hair, for hydrogen and oxygen. And boy, if you're from Texas, we can really tell. Okay? So if you want to commit a crime in California or something, don't leave your hair behind, because we could tell that very easily with these kinds of patternings. And then this just simply shows somebody who volunteered to travel, and then their beard hair was sampled as they moved. And it's the old CSI thing, okay? And CSI stands for Crimes Solved with Isotopes. Okay, thank you very much for your time.
Oh, boy.